Hello, good afternoon, everyone. Let's get started. So we will do a test review today, and if there are things that is not covered in the review that you'd like me to go through, please do let me know anytime, okay? So first question here, label the different layers of the fractionated blood in the diagram below and describe its component. So fractionated blood means you take the blood, you spin it down, and then you're going to separate them based on the mass. So the heavier cells get sink down to the bottom, and then you will have the liquid portion of the blood which stays on the top right here. So what do we call the top portion of the blood here? Anybody remember that? Yeah, that is the plasma. Just give me one moment, please. My pen is not working properly. I just got to fix that. Should be okay now. Okay, yeah, there we go. Okay, so this is the plasma, and we'll label that plasma. Okay, now what is found within the plasma? Well, it is mostly water, right? As you might remember, mostly water with dissolved electrolytes, for example, such as sodium, potassium, chloride, and a bunch of other stuff. There is also going to be dissolved nutrients, wastes, right? And proteins, right? Contains plasma proteins. So now that we have learned quite a bit about plasma proteins in various units, various lessons, does anybody have any examples of plasma proteins that they would like to give me? What are some examples of plasma proteins? Anybody? Yeah, there's definitely antibodies, right? So all those antibodies you make from fighting previous infections, they're going to be there. There are just some general antibodies that are floating around in the plasma. There's also albumin, that's right, which is the major one, right? The most abundant. Plasma proteins. Is there any other proteins that are related, plasma proteins that are related to the immune system other than antibodies? And we can call these like immune protein, right? As in like related to immune system. So antibodies, what else? Adrian said fibrinogen. Those would be like clotting proteins, I would say. Clotting proteins, let's put them down. We have fibrinogen. Okay. Any other examples of immune proteins or another clotting protein? That's okay. So another example of immune protein other than antibody would be complement protein, right? So they are part of your innate immunity. You activate one after the other in a cascade of events. Another clotting proteins other than fibrinogen, we have the prothrombin, right? Okay, so it's a good idea to review the coagulation pathway, where prothrombin is going to get activated into thrombin by thromboplastin, which in turn activate fibrinogen into fibrin, right? There's probably going to be a question where you have to rearrange the orders of events that takes place during coagulation. So make sure you do know that. Okay, and the plasma makes up approximately 55% of your total blood volume. Okay, so there you have it. That's the plasma. The middle layer here, that is your white blood cells. White WBC, white blood cells, plus the platelets. Okay, they don't really have the iron in them, so they're not as heavy. They don't sink down. They just kind of hover in the interface between the plasma and the hematocrit. And that makes up approximately 1% of all your total blood volume. And as you should know by now, there are two classifications for our white cells. We have granulocytes and we have agranulocytes. The marker is so thick, I gotta make it thinner to fit. Examples of granulocytes, anyone? Just give me a bunch. Okay. We have quite a few, right? Yep, I'm seeing some good ones. Neutrophils, basophil, eosinophil. There's one more. That's not introduced in lecture 2, but later on it was introduced like in lecture 3, I believe, and that's also granulocytes, and it's related to basophil. Oh, sorry. Yeah, it is related to basophil. I thought I just gave you the answer. Sorry. Basophil goes around in the body, and then the other one that I'm thinking about, yeah, Maura got it. It's mast cell. That's correct. Okay, so this one is in circulation, right? While this one is like anchored in tissue. Right? They're both responsible for inflammatory response. On the other hand, we have the agranulocytes. So we have the monocytes. Right? We have some lymphocytes such as T cells, B cells. Okay, those are agranulocytes. Okay. Last but not least, we have the RBC over here, the red blood cells. Red blood cells, which forms the hematocrit. Okay, and that's approximately 45% of all your blood volume. Okay, and what do we know for the red cells? I'm just going to give you a few points here. They have no nucleus, for example. Okay, contains hemoglobin, which of course transport oxygen. They have a biconcave shape to maximize surface area. What's another feature that you could think of? Anybody? Yep, lifespan approximately 120 days, and then they will be broken down into at the liver and the spleen, right? Broken down at liver and spleen. Yep, contains hemoglobin. We mentioned that. Very good. And there is a hormone that could stimulate them, right? It's probably worth mentioning it here. EPO, right? You guys remember EPO? Erythropoietin, right? EPO from the kidneys can increase RBC production from the bone marrow. Okay. So remember, all these things, all these blood cells, the white cells, the platelet, the red cells, right? They all came from, oh, that's gonna look messy. Maybe I'll just do it over here. Right. All came from the bone marrow, right? From stem cells. Located in the bone marrow. Okay. The process is called hematopoiesis. Okay. The production of new blood cells, basically. Is that okay? Hope that makes sense. All right. Compare the two test tubes shown below. Explain what might cause the result on the right. So what's the problem here? Okay, this one on the left is the normal one. That's the normal tube. That's what it's supposed to look like when you spin down the blood. You have the plasma here. You have the hematocrit at the bottom. So what's going on with this one? Any suggestions? What if I show you that picture on the test and I ask you, what's a possible explanation? Choose one of the following that best describe what took place. Mihiret said clumping. Could you please expand a little bit? Okay, Tyler said hemolysis. Yeah, both of you are correct, okay? So what we observe here is basically hemolysis, okay? And for those of you who need a refresher on what hemolysis means, it just means that your red cells are bursting open. Okay, so all that hemoglobin, which is what makes the blood red, comes out and it goes into the plasma, making the plasma red instead of yellow. Okay, so why hemolysis? Well, one reason is could be caused by mismatched blood transfusion. I mean, there are other causes as well, okay? Like, but this is the only one that we've learned, right? Okay. Where or when the antibody bind to RBC, a cross reaction occurs, resulting in agglutination, which is clumping, and hemolysis. Okay, if you see observe something like this, is very serious, and you need to figure out the reason, and you need to identify the problem and fix the problem. Okay, otherwise the person might die from this. Okay, internal bleeding can also lead to hemolysis. Okay. All right. Match the parents with their babies. I don't know if you had a chance to. Anybody actually tried this and would like to share their answers? And then I will take it up with the class. Just want to see if anyone has some answers already. What should I put here for the babies? Okay, let's try this. Remember, for something like this, you have to use the 4 rules, right? So AB people cannot have O kids, O cannot have AB. That's okay, Alex. Let's hope this is going to clear it up for you. And 2O will always have O, and 2 negative will always have negative, right? And those are the 4 rules that you should remember when you do these kind of questions. Okay, so it's good to start with people who are like A, B, and O first. That's probably a good starting point, because if the parents is A, B according to the rules I give you, then they cannot have a type O baby, right? So that's not their baby. Okay, if one of the parent is type O, then one of the parent is type O, then their baby cannot be AB. Okay, so there are only 2 choices left. It's either B positive or A negative. But you see, both of the parents are type negative, Rh negative. And if you have 2 negative parents, the baby is guaranteed to be negative. So for that reason, it has to be this baby. Okay, so baby K belongs to this parent, this couple. Does that make sense, the explanation I just gave? Any question, or does anybody need me to repeat that? Okay, so let me repeat one more time. The reason I chose this couple as my starting point is because we have 2 good rules here. We know AB individuals cannot have a type O baby regardless of who their partners is, because if you're AB, you don't have a little I to pass on, right? Similarly, if you're type O, you can only pass on little I, and therefore it's not possible to have an AB child. So if you agree with these 2 statements, then here we can see one of the parents is type AB. So immediately I can reject this as their baby, right? Because AB will not have a type O baby. Similarly, if you're type O, you can only pass on little I. You do not have an IA, you do not have an IB. It's impossible for you to have an AB baby. So that's not yours. Okay, so now we only have 2 other choices left, and we see that both of them are negative. So if both parents are Rh negative, they can only pass on the Rh negative allele, right? And so the baby has to be A negative. Okay, it's not possible for 2 negative parents to have a positive baby, so that's not theirs either. Is that a little bit better? Any follow-up questions? Okay, great. So let's pick another one. Okay, over here, A negative and O negative. We only have 3 babies left. O cannot have AB kids, so that's not theirs. We have 2 negative parents. They cannot have a positive baby, right? And besides, neither of them has a B. It's impossible to have a B anyways. So baby I is going to be theirs. Okay, so baby I is also gone. Okay, next one. I mean, we can work with this one, but I see another O, right? It's always easier to work with parents who has type AB blood or type O blood first, right? So if they have an O parent, AB is not possible. Okay, so that's not theirs, which means baby J is theirs. Okay, and then last but not least, this would be baby H. Okay. Does anyone want me to explain any of these family? Like, you know, how is it possible for A and O to get O? So this must be IA and small I, small I, small I. That's how you end up having an O baby, because it's little I combined with little I, for example. Okay. So no questions? Last call. Okay, thank you. A new blood type called fluffy has been discovered in humans. There are 3 alleles. Sorry, there's a typo here. There are 3 alleles in the fluffy blood group, 1, 2, and 3. If F1 is dominant over F2 and F3, and F2 is dominant over F3, how many different phenotypes are there in the fluffy blood group? So this is something that we talked about in the lecture, right? We had a poll questions or something like that, similar to this. So let's go through it again. So 3 alleles, F1, F2, F3. Let's write down all the possible genotype, all the possible combinations. Okay, so for example, I can have F1 and F1. So I'm just going to write 1 and 1 to avoid the confusion, okay? I'm just going to drop the F. So it could be 1, 1. Let's go 1, 1. Or it could be 2, 2. It could be 3, 3. What other combinations are there besides the 3 that I have written down? Okay, if there are 3 possible allele F1, F2, and F3. So you can have F1, F1, F2, F2, F3, F3. What other combinations exist? Yep, we can have 1, 2, right? An allele from F1 and an allele F2. What else? 1, 3. Yep. 2, 3. That's it, right? So that's all the different possible combinations. If you have 3 alleles, right? Okay. So what would the phenotype be? Okay, let's see. Now, this is easy. If it's 1, 1, then it would be fluffy 1, right? Okay, because there's only 1 allele, right? And then this would be fluffy 2. This would be fluffy 3. Those are the easy one to figure out. But what about this one? What if you have F1 and F2, 1 and 2? Which one shows up? Does 1 show up or does 2 show up? Yeah, people are saying 1 shows up, right? And that makes sense because F1 is dominant over F2, right? So if you have 1 and 2 at the same time, it will still be fluffy 1 because the 1 is the dominant gene. What about 1, 3? Right? Same situation, right? F1 is dominant over both 2 and 3. So when you have 1 and 3, this will also be fluffy 1. Okay, so all the yellow ones there, that's going to be fluffy 1. It doesn't really matter what you call it, okay? I just call it by their name, but this would be 1 blood type. What about 2 and 3? Is 2 and 3 going to be fluffy 2 or fluffy 3? Green or blue? Okay, well, you can read here, right? It says F2 is dominant over F3. Right? So domain is correct. It is going to be fluffy 2. Okay, and we would have fluffy 3. I hope that makes sense. Now, same blood group, but different information now. If all 3 alleles are dominant with co-dominant with each other, how many different phenotypes are there? So let's list the genotypes again. It will be the exact same thing as before. Okay. And so how many phenotypes will there be? Let's do a poll. Okay. Is it 3, 4, 5, 6? Which one is it? Let me launch a poll. Go ahead. Let me know what you think. All right. So we have answers from, all the choices. Let's take a look. Okay. Codominant means both will be expressed. Okay. An example of a codominant situation with the ABO blood group would be AB. If someone is AB blood group, that means they have allele IA and IB, and both of them are going to be expressed. So over here, if you have 1, 1, then that's going to be fluffy 1, fluffy 2, fluffy 3. So that's 3 types already. And if you have 1 and 2, because they are codominant, both of them will be expressed. So you will have both fluffy 1 and fluffy 2 on the surface of the cell. So that's count as a new type of blood, right? Just like AB is neither type A or type B. It's its own like new blood group, right? AB, right? And so that's the 4th one, 5th one, and the 6th one. So the correct answer is D, okay? 6 types. For people who did not choose D, do you understand why it is 6 now? It's okay if you don't understand. You just need to let me know. Let me know what the concern is, and then I will explain to you. All right, because you probably will see something similar to this on the test. Okay. All right. Find the genotype of question mark in the pedigree below. Okay, so who's question mark here? Question mark is this guy. All right. Let's fill in what we know, and then we'll go from there. So to do a pedigree question, we should start with the O's and the ABs, because those are the people where we know for certain what their genotypes are, right? So type O is always little I, little I. There is another type O right here. And AB is always IA, IB. Okay. Over here, we have IB something. We don't know what that something is just yet. We'll come back to it later. And over here, we have IA something and IB something. All right. So let's take a look at question marks. Okay, we have 2 blanks to fill in for each of these individuals. And what we can do is we can look at their children, or we can look at their parents. In this case, it's probably easier to look at their children. Okay, so there's a type O child, girl. So where did she get the little I? Well, one of them has to come from the mother, and then the other one has to come from the father, right? It has to be like that, run from each. So that we'll say this one is from this parent, and then the yellow one is from this parent. Okay. Now we can trace this back for 1 more generation. In this case, this little I probably, well, not probably, definitely came from the grandmother, right? Okay, because the grandfather does not have 1 to pass on. All right. So let's see. Over here, the IA, right? IA something. So where did the IA come from? Well, the IA, is it from the father or from the mother? What do you guys think? Anybody want to tell me? Okay, Adriana said from the mom. Do you want to explain to me why it's from the mom? Or anyone could explain. It doesn't have to be you if you don't feel comfortable unmuting. Why? How do we know it's from the mom and not from the dad?
Can I try?
Yeah, of course, please.
Yeah.
Go ahead.
I think the father's mother and fathers, they are all B types, so they don't have the A type blood. So
Yeah,
the
exactly.
father should be BI. Yes.
Very good.
So
Yeah.
the mom should be AI.
Yep, you got it, right? So if you look at the father and you trace it back 1 more generation, there's absolutely no A in this entire family, right? Okay, so the father here must be, as you said, must be IB. So the IA must have came from here, right? And so the answer is IB little I for the father and IA little I for the mother. Okay, that is that. Again, I hope it makes sense. And if it doesn't, well, maybe think about it a little bit more, and then feel free to contact me. There's still one more day, right, before the test. So, you know, if you need some one-on-one or something, let me know. I can have a session with you tomorrow if you like. All right, this is a multiple choice question. I don't know if you had a chance to try that, but I will launch the poll and let you pick the answer. Yeah, today is the last class with me. Next week, Miren will take over. By the way, I just updated the weekly schedule. Miren has filled in his session, his half of the course. So you guys can go ahead and get the schedule on eCentennial later on. It's pretty much the same format. He's gonna have asynchronous followed by synchronous sessions. So not much change in that regard. Okay, so I know this one's a little bit wordy, and you know, you might still be reading about it, but I think we have good response so far. So I'm going to end the poll, and you see that again there are kind of answers everywhere, which is good. That means it's a good question to take up. So let's take a look at what's going on. So Sarah's blood was mixed with antibody A. So it's like those questions, right, from the lab, right? Like, you know, you're gonna have 3 wells or something, and then this is gonna be the A, the B, and then the O. And the drop of blood is mixed with anti-A, B, and then RH. Oh, RH here, sorry. Clumping is not seen in any of the tests. So no clumping here, no clumping here, no clumping here. What does it mean? A, the test must have been defective because clumping should have been seen in at least one test. That's not necessarily true. If you don't have A, if you don't have B, and you don't have RH, then no clumping would occur, right? In order for clumping to occur, it means you have to have that antigen, right? And it's okay to not have A, not have B, and not have RH. So this is not true, okay? You don't need to have at least one test having clumping. All right, you can have all of them clumping, you can have none of them clumping, and anything in between. Okay, none of the antibodies in Sarah's plasma reacted with the antibodies. So this is not correct, because when we mix the blood, when we test the blood this way, we're not checking to see if the antibodies are reacting with the antibodies. Okay, we are checking whether the antigens are reacting with the antibodies. Right, the antigens are the stuff that are on the RBC, right? We want to see if those will be recognized by the antibodies. So it has nothing to do with Sarah's antibodies at all in this particular case. Sarah's blood type is O positive. This is the most popular choice, but that is not correct either, because if she was O positive, you would have seen clumping in the RH, right? Because no clumping was observed in the RH. That means she does not have the RH blood group on her red blood cell, and so she must be negative. In fact, she must be O negative, right? If you think about it, no A, no B means O, and no reaction with RH means negative. Okay, so she is O negative. And that makes this the correct choice. Okay, Sarah does not have any ABO or RH antigens on her RBC, which is why she didn't have any reactions, right, in any of the wells. Is that okay? Now, don't worry, not all the questions are going to be this complicated. All right. Once more for you. Okay. Do you have a specific question, Alex, like in terms of which part can, like, do you understand the concept of how to test work? Right. So like maybe we go back to reaction with RH. Okay. So like look at these stuff, right? And this is from lecture 2. When you do see the clumping, like these dots, that tells you your cell has that antigen. Okay, so the fact that there's no reaction with A or RH, that means there is no A or RH on the RBC, right? So on the, if you draw the red cells, right, we do not draw A because they don't have it. We can draw B because you see the clumping, and then we don't draw RH. So if you have a red cell that only has B on it, that's B negative, right? Okay. Let's see. We don't have an O negative here, but let's take a look at this one. Okay, this one, again, we do the red cell here, and no reaction with A, so we do not put the letter A on the red cell. We do not put the letter B on the red cell, but there is clumping in RH here, so we're going to put RH on it. So with no A and no B, it's going to be type O. And if someone has RH, we call them positive. All right. Yeah, that was a good video in the lab one. So if you haven't had a chance to watch that, please do so. Normally it would even be better if you're able to do this hands-on yourself. It really helps with the understanding. But yeah, unfortunately we couldn't. So back to here, right? Like let's just draw our happy face, eyeless happy face right here. No A, no B, so I do not put those letters on. No RH, we do not put the RH on either. So if you don't have AB, you're O. If you don't have RH, you're negative. Right? So that's why we said she does not have ABO or RH. Okay. Yeah, that's her phenotype, exactly. Very good. Okay, select all the safe blood transfusion. Let's do that. Let's do that. This one is multi-select. Go ahead. What is Sarah's blood type? O negative, right? O negative. Because there's no A, no B. Yeah. Okay, I'm gonna wrap this up. If you could lock in your answers, that would be great. Okay, well, thank you for participating. Here we go. O positive to A negative, that is not safe. Positive cannot be given to negative, right? Okay, we're gonna sensitize the person, or if they previously had been sensitized, you're gonna cause hemolysis. So not good. Positive cannot be given to negative. AB negative to AB positive, that's okay. That's okay. So B is the correct answer. A positive to AB positive, that is okay. Right? Think about the transfusion. I don't know what you call this, transfusion flow chart, right? A can give to AB, right? And positive to positive is okay. B negative to AB negative. Negative can be given to negative, and B can be given to AB, so that's okay as well. O positive to O negative is not okay for the same reason, right? Positive should not be given to negative. Okay. Is any of these not clear? Anyone need me to take up any of these? Okay. This is just a table that has all like a lot of the cells in it, and it's just a good place to, put them all together. And we'll fill that in. All right. So macrophages, right? So the macrophages, they are actually differentiated from monocytes. Okay, so the monocytes like kind of patrol around the body, and then when they see something that's not supposed to be there, they transform, they differentiate into these macrophage, right? And how do we classify macrophage? Well, this is the jack of all trades, as they call it. Okay, there is the phagocyte. They eat stuff, so we call them phagocytes. They are not only can they eat them, they could eat them and then put them on the surface and go activate the naive cell. So we call them an antigen presenting cell, right? Or an APC for short. Right? We can also classify macrophage as an agranulocyte. All right. What do? Well, they go around and eat stuff, right? They will perform phagocytosis, right? On anything with PAMs, for example, the pathogen associated molecular patterns. They are a group of molecules that the macrophage recognizes as something that's dangerous, something that's not supposed to be in the body. Okay, they could do antigen processing by putting the whatever they eat on their surface plus antigen presentation, showing it to the naive cells, for example. And of course, they can activate naive T cells. There you go. Okay. Next, we have the neutrophils. We can classify them as granulocytes. Right? They are also phagocytes. They can also eat stuff up. It's just that they cannot present it. Okay, so they're not APC. The main function is just phagocytosis. Right? Walk around, eat things up. I don't know if I told you, but the child of one of my friends, when he was little, he didn't have enough neutrophils, so he wasn't allowed to go to daycare, right? Because neutrophils, as you know, is one of the most abundant white cells, and they do a lot of the cleaning up, right? And so if you don't have enough, you're more susceptible to getting infection. But as he grew up, the count went back to normal, and yeah, he could go to school now. Okay, mast cells and basophils, they are granulocytes. Okay, so the difference is basophil is in circulation, and then the mast cells, they are anchored to tissue. Their jobs are similar. They initiate inflammation. Right, by releasing histamine. Okay. The main difference is that the mast cells is more for localized type of inflammation, Again, like when you cut your finger or something. And then basophil, that's more for like systemic, right? We can have mild situations like allergies where it's not life-threatening. It's a nuisance though. And it could go all the way to things like peanut allergy, right? Then that would be very dangerous systemic inflammation. Okay, next we have the NK cells, which stands for natural killer cells, and we classify them as a part of the innate immunity. And their job is to recognize infected cells and cancerous cells. Okay. Helper T cells. This is part of cell-mediated immunity. Cell-mediated, and it's the adaptive branch. Okay. And the helper T cell is going to help activate naive B cells. Right? They will help coordinate immune response. Plasma cells, they are part of, instead of cell-mediated, we have antibody mediated adaptive immunity. And their main function is to mass produce antibodies. Okay. Any questions about any of the cells? Okay. Thank you. Number 9, how could you contrast specific and non-specific immunity? All right, let's do the poll. Go ahead. Okay, gonna end the poll there. Let's see. So just for the record, specific defense is referring to adaptive immunity, right? So you're gonna have the antibody production, you're gonna have high specificity, you're going to have immune memory. It is not going to be immediate, right? Those are examples of specific defense. Whereas non-specific defense are things that you're born with. As soon as you come into this world, you have them. That's the inflammation, surface barriers, complement proteins, fever, so on and so forth. Okay, so how are they different? Well, first one, specific immunity works only after non-specific defense have failed. That's not true. The two branch can happen simultaneously. Doesn't have to be one after the other. Non-specific defense and specific immunity requires a prior exposure to be effective. Not true, right? Non-specific defense, in fact, does not require prior exposure at all. It is just going to work immediately. Phagocytosis, for example, if there's a cell that has PAMs on it, you don't need to see that before in order to eat it, right? You're just going to eat it right away. B cells work on both non-specific and specific immunity. That's not correct. B and T cells are exclusive for specific immunity, right? Specific immunity changes throughout a person's life, but non-specific defense does not. That's correct, okay? So things like the ability to have a fever, inflammation, all those mechanisms does not change. Your surface barriers don't change, right, throughout your life. But your specific immunity changes. Every time you encounter a new pathogen and you fight it off, you're going to be able to have new memory cells, and that change your specific immunity. Okay, let's take a look at this one. We'll do together. Which of the following statements describe a form of acquired immunity? Okay, so acquired immunity is another word for adaptive, right? Adaptive immunity. Right? It's another word for specific immunity, right? Okay. So a RhoGAM injection is an example of artificial active immunity. Okay, is that correct? Well, no, because giving you RhoGAM does not give you protection against anything, right? We in fact we don't use RhoGAM for protection, right? We use RhoGAM to prevent sensitization, right? So A and B are both nonsense. Okay, RhoGAMs is nothing to do with passive or active immunity. Okay, nursing an infant to provide the antibodies in breast milk is an example of natural active immunity. That's not correct. It's supposed to be passive because the antibodies are transferred, right? The host, the baby, is not going to be the one who's making the antibodies. So that's why it's passive and not active. Catching COVID-19 and recovering from it should give you natural active immunity. That is the correct answer. Okay, if you naturally caught the disease, then it's natural. Okay, like nobody's going to deliberately get infected by it. Active because you have to make your own antibody, right? So that's the correct one. And let's just check the last one. Receiving an mRNA COVID vaccine is an example of getting artificial passive immunity. So that's artificial is correct, but not passive, right? Getting the vaccine is just going to introduce the antigen to your system. You still have to make the antibody yourself. Okay, so that's why D is the correct answer. Okay. Name the cell. All right, let's see what this is. Use perforin to put holes in infected cells. That would be the killer T cell. There's a slide on killer T cell. Another name for killer T cell is cytotoxic T cell. Okay, and they put holes in their target. Okay? The thing that put holes in it is called perforin, okay? As in they perforate the membrane. Release histamine to initiate local inflammatory response. You should know that by now. It is mast cell. A type of APC that activates naive T cells. We only learn one type, which is the macrophage. Kills cancerous cells. That would be NK cells. You want them to do their job. forms blood clot. That would be the function of platelets. And the most abundant WBC, that would be neutrophil. How about MAC? Okay, so MAC is the membrane attack complex. That's not used by T cells. Okay, this is a part of complement proteins. Right, complement proteins. Right. So that also punch holes in the bacteria, but it's the protein that does that. Okay, it's this is a different type of different type of proteins. Okay, this protein is secreted by the killer T cells. This is the end product of all those reactions that takes place when you activate complement proteins. Furthermore, this is part of innate immunity, whereas this thing that we're describing here, because it involves T cells, is part of adaptive immunity. Albumin is not a WBC. Albumin is a plasma protein. Okay, so it's just one of the many proteins that exist in the yellow portion, the liquid portion of the blood, and it's the most abundant one. Okay, you might remember we mentioned in the lecture that the albumin, because you have a lot of them in the blood, it helps draw in the blood, right? Okay, it helps maintain osmotic pressure, right? That's what the albumin is for. Okay. All right, give examples for each of the following. Formed elements. Okay, formed elements is just another way to say blood cells. I don't know why they can't just call it blood cells. Why do they have to make it so complicated? I do not know, but here they are. Red cells, white cells, and platelets. Okay. And just in case, I honestly don't remember if we have these words in the test, but RBC is erythrocytes. Okay, white cells is leukocytes. I think they should be in the lecture notes, right? And this is thrombocytes. Just in case you see them. Oh, and I got an email from a student saying that there are some weird questions in the self quiz about the animal respiratory system. So we did not learn those in the lessons. So you can just ignore them. You will not be responsible for those questions. If you haven't tried the self quiz yet, you don't have to worry about it because I already deleted the question. But I saw that a couple of you have tried it, and in case you saw those animal respiratory questions and it worries you, don't worry, it won't be on. All right. Granulocytes. We did this a couple times already. So neutrophils, basophils, and the eosinophil for fighting parasites. Next, we have the lymphocytes, which is T cells and B cells. And of course, there are different types, right? The different effectors that we could get. Coagulation proteins, we mentioned earlier, there is prothrombin, there is fibrinogen. Okay, these are just some examples that we did, right? By no means this is these are the only examples, right? Anybody heard of hemophilia by any chance? Someone is a hemophiliac. What does it mean? Anybody heard of that before? Not so scared, not so much scared of blood, but their blood don't clot, right, easily. They do have platelets, but it's just that their blood is not clotting quick enough. Okay, so like if they have a small cut, it could take a very long time before it stops bleeding, and sometimes if you have more serious cut, whereas a normal person would be able to apply pressure and stop the bleeding, that person might have to go to the hospital to get the blood clot to form. So those people have mutations in various clotting proteins in the body. Okay, so these are just two of them, but there are many other clotting proteins, right? So some are hereditary disease related. If you mutate those, you can have a hemophilia. Just some extra information. It's not on the test, okay? I just like to tell you things as they come into my mind. Not quite similar to diabetics. So diabetics is a completely different mechanism in terms of the the capillaries being leaky and all that, and the swelling. That's a different reason, not because of clotting factors. Okay, autoimmune disease. Anybody have any autoimmune disease that they remember from the lesson or that they know of? Yep, lupus. That's a good one. Anyone else? Multiple sclerosis. Yeah. ALS, I believe. Is that what you mean, Myriad? ALS. Yeah. Oh, that's okay. Yeah. Addison's disease and a bunch of other ones that you probably know. Okay, but here are some examples. Physical barriers. We have intact skin, right? We have mucous membrane, right? Normal flora. Immunodeficiency, we only learned two types, AIDS and SCID, right? SCID is the one that there's a mutation and the baby is born without any immunity at all, right? They live in a bubble, the bubble boy syndrome, so-called. Lymphatic organs, we got a bunch. Spleen, tonsils, right? Thymus. Artificial passive immunity. An example would be serum. I remember that question about Joffrey getting bit by a snake, and then he would need a serum at the hospital, right? Me getting bit by a dog, I also needed serum at the doctor's office. Natural passive immunity, that would be breastfeeding. Breastfeeding. There you go. Okay, characteristics of inflammation: redness, swelling, pain, warmth. These are side effects of histamine, basically. Okay. I think we did these questions already during the poll, so we're not gonna go through them again. I will just circle the answer for you, right? B here and B here. Okay, we did this already in the polls during that lecture. Okay, number 15. Answer each of the following question based on the graph shown. Okay, so this is the breathing graph, as you know, right? Your tidal volume, so on and so forth. What is the expiratory reserve volume for this person? So the expiratory reserve volume is the volume of air that you can exhale, right? After a regular exhalation. So how much more air can you breathe out after your normal breathing out, so to speak, right? So that is the expiratory reserve volume right here, this part right here. Okay, so we can see we'll say this is about 2400 maybe, and then this one here is about about 1400. So if you subtract them, you would have about 1000 milliliters. And I know what you're thinking. You're like, well, what if I get this question on the test? These numbers seem so arbitrary, right? Like it seems so random. What if I think this is like 2300? Would I get it wrong? Don't worry. The question on the test will be very clear. Okay, there will be no guessing involved. Okay, it's going to be very easy to tell what number it is. It's just that there isn't a lot of these graphs that I can find, and the ones that I'm able to find, sometimes they don't work out perfectly. But the key thing is you have to know what is the expiratory reserve volume. All right. What is labeled as B? What do you think? What is this thing right here? B, from here all the way. That is the vital capacity, correct? Okay, vital capacity is, once again, inspiratory reserve volume, which is C, plus the tidal volume, which is this thing that goes hover up and down, as well as expiratory reserve volume. Remember, your vital capacity will not change, even if you change your physical state of activity. Okay, whether you're lying down or running around, doing a marathon, it doesn't matter. Your vital capacity remains the same, but these things might change accordingly. If one goes up, the other must go down to maintain the same number. Okay, describe the breathing pattern for the person. Well, just normal breathing here, right? And then maximal exhalation, and then go back to normal breathing, and then maximum inhalation, basically, right? And then go back to normal breathing, breathe in as much as you can, and then exhale all the way until you cannot exhale anymore, and then normal breathing. Okay, should make sense. Describe the volume label as A. A is the, oh, this A I'm talking about, not that A, sorry. This A, okay? So that is the dead volume, right? The residual volume. Okay, we talk about what the residual volume is. It is very concentrated, has very high concentration of carbon dioxide, right? It is not involved in gas exchange. We do not want too much residual volume, okay? But if you have emphysema, right, where the lung loses its elasticity, then the residual volume is going to go up. Okay, moving on. Draw a flowchart to illustrate how inhalation and exhalation occurs. I think in the lecture we did inhalation. Oops, the whole thing is gone. I don't know why, but I believe we did inhalation. So I'm going to do exhalation for a change. All right, so this is the diagram that we have. Okay, so for exhalation, you want the diaphragm to resume the dome shape. So it's going to go up, and at the same time, the rib cage is going to come down. Okay, so collectively, this is going to squeeze on the lungs, right? So rib cage comes down, diaphragm goes up, and you squeeze on the lungs. Okay, so both. We say rib cage, relax, relaxes, I guess, resumes dome shape, right? Say moves up, and then at the same time, rib cage. No, I don't mean rib cage. I mean diaphragm. I'm sorry. Diaphragm. Okay, and then the rib cage moves inwards. Okay, comes in, comes down. So collectively, these action will decrease lung volume, which would then increase pulmonary pressure, right? So this is Boyle's law, which states that at constant temperature, when volume goes up, pressure goes down. And then when volume goes down, pressure goes up. Okay. So once PI is greater than PA, atmospheric pressure, then we have exhalation. Okay. So the exhalation is facilitated by the elasticity of the lungs, right? Okay, the stretchiness of the lungs, and in part that's due to the surface tension created by the moisture around the alveoli. Okay. Couple more. I'll just take these up with you. During swallowing, which of the following occurs? The larynx moves upward against the epiglottis. The epiglottis moves downwards to seal the esophagus. Pharynx moves upward against the epiglottis. The trachea moves upward against the epiglottis. Okay, so epiglottis itself does not move, so that's not correct. Okay, the larynx is the one that moves upwards against the epiglottis. Okay, these guys don't actually move. The pharynx is the back of your throat, right? How can the back of your throat move up? It's just like the back of the throat is or like your throat is your throat, right? It doesn't move up or down, right? It's the larynx, the voice box, right? You can feel it. Right? Put your hand here and swallow, you can feel it move up. Surfactant, that's the oily stuff that is secreted by septal cells, right? Oily substances secreted by septal cells. Okay, and the purpose is to break the surface tension. By breaking the surface tension, you increase the, so I'll write down here, break surface tension at alveoli. Right, this would increase compliance, the expandability of the lungs. Okay, so by increasing compliance, you will allow inhalation. Okay, the urge to breathe is primarily driven by, I hope you know by now, it is the increase of carbon dioxide in the blood. Okay, this will be detected by the medulla oblongata, which is part of the brainstem, right? You have a respiratory center there that detects the amount of carbon dioxide in your blood. Okay. I think there are a couple of other good poll questions from last time that we didn't have time to go through. Oh, there's only one good question that we didn't go through. It's probably worth talking about. Let me launch the poll for you. And let's go through that. Okay, I'll give you a little bit more time. Okay, let's take this up. Dawn is not able to produce surfactant. I just told you what surfactant does. In order to inhale normal tidal volume, which of the following must happen? A, she will need to inhale more forcefully to lower intrapulmonary pressure. Okay, maybe. She will need to exhale more forcefully to empty her lungs at the end of each breath. So what does surfactant do? Well, I told you, right? Makes it easier to inhale, right? By increasing the compliance. Okay, so if you don't have that, it is going to affect, if you don't have enough of that, or if you don't produce that at all, it's going to affect your inhalation. It's not going to affect your exhalation, right? So A, between A and B, A is the correct one. B is not correct. But let's just double check the other ones. She will need to breathe air with higher oxygen content. The problem is not with oxygen content, right? The problem is to get the air into her system in the first place, right? So having more oxygen alone will not fix the problem, right? She needs to be able to get it in to her lungs. She can just breathe normally as surfactant. That's not true. Surfactant definitely affects your tidal volume for the reason I told you about. So the answer is A. Okay, she will need to forcefully inhale more forcefully to lower the intrapulmonary pressure so that she can bring in the air and force the lung to expand. Okay, that is it. So like I said, this is the last class with me. You can still email me. I will still be available next week as well. If you have any problems related to the course or whatever, you can still find me. But then I will no longer be. I'll be on vacation starting the 20th, so you won't be able to reach me. So if you need anything else, please do make sure to email me before then. And like I said, the new schedule is available now on eCentennial. I'll place a link for you. And Professor Moran is going to take over starting next week. Okay, so test on Thursday. Make sure you finish your lab quiz today, right? That's the timeline, deadline. And if you need extra help before the test, feel free to email me. We can set up something for tomorrow. Okay. Other than that, well, thank you so much for attending the tutorial all the time. I recognize almost all of your names now, and I wish you success for the rest of the course, the rest of the semester. And if you ever need reference or anything, feel free to reach out. I'll be happy to provide what you need. Okay. Okay. Thank you. Bye now.